Hi, I'm Sebastian Dupraz. I'm a senior scientist in the Institute for Neurovascular Cell Biology, where I work to understand the role and function of the neurovascular unit in central nervous system development and disease. I'm the co-first author of the work Centrosomal microtubule nucleation regulates radial migration of projection neurons independently of polarization in the developing brain. And in the next 2 minutes, I'll tell you what it's all about. Our work demonstrated that neuronal migration and axon formation, two intertwined processes, are actually driven by independent microtubule machineries during cortical development. This work provides the scientific community with a conceptual advance. This is neurons migration and axon formation, two intertwined processes that share common components are mechanistically separable. This will help us to design better experimental paradigms to interrogate these functions and therefore to better understand the molecules and nature of the pathology that are derived from their malfunction. Apart from the conceptual advance, a very important aspect of this work is the generation of very precise molecular tools. We use these tools to modulate a very particular function of the centrosome. This is the microtubule nucleation activity without having to take out the whole organelle of the game. This really allows us to address a particular function of the centrosome and understand its neurodevelopmental role with unprecedented accuracy. This one definitely has to be figure number 6. This figure shows how one of our molecular tools effectively suppresses microtubule nucleating activity in the centrosome in vivo, impairing neuronal migration but not axon formation. Scientifically, I like that figure because it shows the conversion of three independent lines of work, namely the production in vitro of the molecular tools, the design of the molecular method of delivery, and the work in vivo per se. And personally, I really like it because it shows us that these three different lines of work, they came together exactly as we envisioned them in the drawing board. I can still recall how excited I was in the microscope while I was scanning these images. At the moment, we have a good understanding of the molecules and mechanisms orchestrating centrosomal microtubule nucleation that drive neural migration. We hope that by fine-tuning our molecular tools, we will also be able to focus on the identity and roles of those molecules that regulate acentrosomal microtubule nucleation, which our work shows is the engine driving axon work, and naturally explore the relevance of a centrosomal microtubule nucleation for axon regeneration.